Good afternoon. Before we begin, a few reminders. Please keep your cameras off and microphones muted so as to not distract the speakers and other participants. We encourage everyone to actively participate by asking questions. Just write them on the chat box and we will try to get to as many of them as we can after the three speakers have presented. So we hope that you will stay until after all three lectures have been delivered. At the end of the webinar, we will provide the link to the evaluation form that you must fill out in order to receive your certificate of attendance. Lastly, there will be a photo documentation session at the end. The photos may be posted online, so for data privacy reasons, if you do not consent to have your image captured, please keep your cameras off during the photo session and remove your profile pictures. If you also prefer to not have even your name appear in the photos, you may choose to leave the meeting at this point before the photo documentation. This webinar will be recorded, so those who are unable to attend the webinar today or who would like to watch the lectures again may view the video which will be posted on the BRSL Facebook page. We are also streaming live on YouTube, so you may watch the webinar on the BRSL YouTube channel. And with that, let's begin. Welcome to the third installment of the BRSL
Hello everyone, good afternoon. I'm sorry for the inconvenience for the internet interruption. Again, we would like to welcome you everyone to the third installment of the BRSL webinar series with the theme Complexly Microbial. So to start off this session, let us first hear a few words from the head of the BRSL, Dr. Pierre Angelo Vital. Ma'am.
Thank you, Jessa and Erna. Good afternoon, everyone. I formally welcome you to the third of the Biological Research and Services Laboratory of Natural Sciences Research Institute, University of the Philippines, Diliman, or simply BRSL webinar series titled Complexly Microbial Episode 3, a webinar series on microbial life as part of the efforts of BRSL to contribute to the understanding of science, particularly biology, in the community. Welcome back to those who have been with us in the last two episodes. For this webinar, we are fortunate to be joined by experts Dr. Marie-Christine Obusan, a good friend and colleague from the Institute of Biology, University of the Philippines, Diliman, with topic of talk in the field of environmental science, Assistant Professor Francis Ligario from the Institute of Aquaculture, University of Stirling, Scotland, with topic of talk on aquatic pathobiology, and our very own Ms. Joy Ann Santos from BRSL NSRI UP Diliman with interesting topic on molecular microbiology and who is also involved in COVID diagnostic kit development. They will walk us through their minute but significant worlds, and as participants, you will be given the chance to interact with them through our Q&A session later. This webinar will not be possible without the support from our NSRI Director, Dr. Jonas Kilang, and the efforts of the amazing and hardworking researchers and staff of BRSL. And for this webinar, your co-hosts, as you can see from your participants tab, are Rian, Brian, Jude, Jessa, Donna, Serena, Ryan May, Raquel, Erna. Credit, of course, goes to them. On another note, you might also be interested once we start the new normal of everyone going out. So please allow me to share this PowerPoint presentation to further introduce our laboratory. I hope you can see the screen now.
Yes, ma'am.
Thank you. So for the first slide, it is a facilities available in BRSL. And these facilities include the DNA laboratory, the cell and tissue culture lab, the microbiology lab, microscope room, as well as the general equipment lab. The next slide is, of course, on the researches done by our lab under different project leaders. And these are natural products, genotoxicity of environmental pollutants, bioarchaeology, with impact on food safety and sustainable or renewable energy, phycology, virology, molecular diagnostics, and biorisk assessment. The next would be the different workshops that we offer, which include Advanced Molecular Biology Techniques Workshop, Natural Products Workshop, the Basic Molecular Biology Techniques Workshop, Plant Chromosome Preparation Workshop, the Microalgae or the Phycology Workshop, as well as the Laboratory Animal Care and Use, which is in compliance with the Republic Act 8485, or famously known as the Animal Welfare Act. And then fourth slide is on technical services, which include technical advising and online consultation for this pandemic, research assistance and internship as soon as we are back in the lab, lab and equipment use, microscopy, bioassays, as well as this webinar and lecture series. And so I just would like to share with you lastly our group picture during one of the workshops we conducted. So please feel free to email or contact us if you have biological concerns, and do not forget to like our Facebook page. So now please allow me to end my screen share, as I also would like to share with you a video presentation of the post-enablement session on SAP data analytics with corresponding information on ASEAN Data Science Explorers competition. As part of BRSL's efforts to have a greater international reach, BRSL has been a co-organizer of this enablement session, together with Computational Science Research Center of UP Diliman and the ASEAN Foundation based in Jakarta, Indonesia. Although competition for 2020 is over, I still would like to invite you to consider learning data analytics, especially during this time of pandemic, where most of us migrated to online work, and later on participate in the next competition for 2021. At the end, we will share the evaluation link in the chat box. You may fill it up after our webinar, and please give us at least 2 weeks to send your certificate for completing the post-enablement video watching. Note that this is a separate certificate from the certificate to be provided at the end of this webinar. So Brian, if you can kindly share the video, so please enjoy watching.
Down? Wanted to do something worthwhile during this quarantine season? Well, you are on the right page. Let me introduce to you the ASEAN Data Science Explorers 2020 competition. The ASEAN Data Science Explorers, or the ADSE, is a flagship program run by the ASEAN Foundation and SAP to help unlock the youth's potential and create a positive social impact in Southeast Asia. It is a competition to showcase the youth's digital literacy and capacity to create data-driven proposals to develop solutions towards ASEAN growth and development to improve people's lives. The proposals highlight pressing social and economic issues in ASEAN across the 6 selected United Nations Sustainable Development Goals. The platform to be used for the competition is the SAP Data Analytics Cloud. SAP Data Analytics Cloud is a tool for data exploration, visualization and analysis, planning, and predictive forecasting. Using the SAP Analytics Cloud, you can start analyzing your dataset and create a report or story. In this video, I am going to use a dataset on UK accident information and ask the SAP Analytics Cloud to figure out what are the probable influencers of number of casualties in these accidents. First, import your dataset into the SAP Data Analytics Cloud by clicking the menu button. Click Create Model. Then, click Import a file from your computer. Click Select Sourced File. Then, select your dataset. Once you have uploaded your dataset, you can also check if your data is accurate or clean by changing the view into Card View. If these are all in green, it means that you are good to go. You can also go back to Grid View so that you can edit the errors in your dataset. For more details on data modeling, you may check this video. Now that you have already uploaded your data, validated the data and created the model, you are now ready to create a story. A story is what we call a form of reports and visualization on SAP Analytics Cloud. To start, click the menu button. Click Create Story. With SAP Data Analytics Cloud, you have several options to start from using some of templates here at the bottom right. And now, we can use the Smart Discovery feature, which will allow you to uncover new or unknown relationships in your data. Click Run a Smart Discovery. Select the data set you have uploaded. Next, click Discovery Settings. Select a measure dimension. For example, choose Number of Accidents, then click Run. SAP Data Analytics will then generate your story. So, in just a few clicks, we are able to generate all of these charts automatically, so the results of a Smart Discovery makes for a really good starting point when it comes to creating charts. You can further navigate around and dig into the details of your data analysis. Now, let's move on to another important feature of SAP, the simulation feature. A simulation ties together all the different influencers we were able to pull together. It helps us to forecast a result if there were changes in the influencers. For example, we will tell if the number of casualties will increase or decrease depending on the following factors: time of the day, and road conditions. Click Simulate. Now, we can foretell that if accidents occur in the evening and during wet road conditions, this will result to an increased number of casualties. Easy, right? Smart Discovery is just one of the many features you can explore with SAP Data Analytics. You can analyze various forms of data as well as generate different stories. Now, using the SAP Data Analytics Cloud, you can join the ASEAN Data Science Explorers 2020 competition. Simply come up with data analytics on socio-economic issues and data analytics-based solutions or recommendations to solve the issues. Submit your entry based on one of these 6 categories, and come up with practical ideas that can contribute to the achievement of both the ASEAN Economic Community Blueprint 2025, and the ASEAN Socio-Cultural Community Blueprint 2025. These 6 goals are also part of the United Nations Sustainable Development Goals. Prizes are at stake. Plus, this would also be the best avenue for you to improve your analytical and critical thinking skills, organizational skills, decision-making and problem-solving skills, paying attention to details, as well as gathering, viewing, and analyzing different forms of information in details. For more details on the ASEAN Data Science Explorers 2020 competition, as well as how to use the SAP Data Analytics Cloud, simply visit aseandse.org or contact the administrator of this page.
Thank you so much, Brian, for sharing the video. For those of you who would like to watch it again, we will paste the link in the chat box as well as again the evaluation form. As an advanced announcement next month, we will be partnering with the Philippine Phycological Society Incorporated for our webinar. So please stay tuned with our Facebook page for updates. With this, I wish you to have a productive webinar, and with our great speakers for this afternoon, I'm very sure you will. Thank you very much, and I turn you back over to Jessa and Erna.
Thank you, Dr. Vital. And now, on to our first speaker. Our first speaker is one of our researchers. Ms. Joy Ann Santos is a university researcher at the BRSL. She finished her master's degree in microbiology from the Institute of Biology, UP Diliman, where she is also currently in the PhD in Biology program, specializing in microbiology and molecular biology. She has been and is presently involved in numerous research projects in the field of molecular diagnostics of pathogens, most notably in the detection of Schistosoma japonicum, Vibrio parahemolyticus, Salmonella enterica, chikungunya virus, dengue virus, and most recently enteric viruses in the environment and coronavirus disease 2019 or COVID-19. She is part of the team at the Manila Health Tech, which developed the local COVID-19 RT-PCR detection kit. But today, she will share with us a little bit from her extensive work on dengue virus detection with her talk entitled Early Detection of Dengue Virus Infections Through Local Innovations. Please welcome Ms. Joy Ann Santos.
Good afternoon, everyone. Can you see my slides?
Yes, Miss Chaps.
Thank you. So, again, good afternoon. Today, I will share with you one of the um, most successful technologies that have been commercialized or is already available in the market today. So, this technology started from the lab, which ended to its end user. The outline of my talk for today. Uh, this one. So I will shortly discuss about dengue virus and dengue infection, then describe the different diagnostic techniques being done to detect dengue infections, aqua kit that we have developed, and the implications of providing and developing local technologies. So the dengue virus belongs to the family Flaviviridae, a family of positive, single-stranded, enveloped RNA viruses. So they are found in arthropods, primarily ticks and mosquitoes, and can occasionally infect humans. Members of this family belong to a single genus, the flavivirus, and cause widespread morbidity and mortality throughout the world. There are four serotypes of the dengue virus: dengue 1, 2, 3, and 4. Primary infection with any serotype confers lifelong protection against the homologous serotype, but no protection against the secondary infection with a heterologous serotype. This only means that pag na-infect ka, kunyari ng dengue serotype 1, you already have protection for that specific serotype. Pero wala ka pa ding protection with the other three. So humans are the main reservoir for this virus. And dengue is considered the most important viral infection transmitted among humans by arthropods. Dengue transmission is primarily by Aedes mosquitoes. Aedes aegypti being the principal vector, or the secondary one is the Albopictus species. They feed preferentially on human blood, and they are a daytime feeder. They they have imperceptible bite. Ibig sabihin, hindi mo mararamdaman na kinagat ka na pala ng lamok. And they are very well adapted to the life in the urban settings because actually, medyo maarte sila. They breed in clean, stagnant water in containers that collected rainwater. That's why, as part of the DOH campaign to prevent yung outbreak ng dengue is yung pag-clean ng ating environment. So, importante din yun to maintain cleanliness in the environment para to prevent the spread of these mosquitoes na vectors of the virus. So mild symptoms of dengue can be confused with other illnesses that cause fever, aches, and pains or rash. The most common symptom of dengue fever is with any of the following: nausea, vomiting, rash, aches and pains, eye pain typically behind your eyes, muscle, joint, or bone pain. So, as you can see, ito yung symptoms ng flu. So, if you don't detect the disease, pwede mo siyang ma-mistaken na normal flu lang. About 1 in 20 people who get sick with dengue will develop severe dengue. Severe dengue is a more serious form of disease that can result in shock, internal bleeding, and even death. There are different clinical presentations in a dengue patient, which ranges from the mild dengue fever, and then dengue without warning signs, dengue with warning signs, severe dengue, and dengue with organ failure. Usually, this is the one that causes death in a patient, especially if the disease is not detected at an early stage. The incubation period of the virus is approximately 18 days. So, after you have been bitten by the mosquito, it will take around 18 days before you start to have fever. So, your viral load will start to plummet from day zero or the onset of your fever. So, yung day kung kailan ka nagkalagnat, yun yung day zero mo. And will last up to 5 days, first 5 days, and will start to drop after that. So on day 7 onwards, you'll develop antibodies now. Studies have shown that dengue intervention given at an early stage or early onset of the disease, usually the first 5 days, can be life saving. So, if the infection is detected at an early stage, there would be better outcomes because uh, the clinician or the attending physician will be able to manage the patient properly at a very uh, early stage. So hindi na kayang mag-progress into the severe stage. So, however, current diagnostic modalities vary in terms of sensitivity and technology availability. In most cases, we settle for the rapid technology platforms like the Dengue NS1, yung mga RDTs, that provide sensitivity that is heavily affected by the patient's immune status. However, nucleic acid based, like the RT-PCR, are the most sensitive or the best tool that can circumvent a patient's immune status, but it's very uh, uh, costly to the patient. So therefore, limits the access of most Filipinos. So there are different diagnostic techniques being used for dengue diagnosis. So there are two basic methods for establishing a diagnosis. The first one is the detection of the virus itself, which is the indirect method, and the detection of the anti-dengue antibodies, which are the indirect methods. So direct methods include the virus isolation or the viral culture, genome detection, like PCR or other nucleic acid amplification tests, and antigen detection like your NS1. Indirect methods include the serology, uh, like ELISA for your IgM and IgG. So direct methods are more specific, but uh, not all not all patients have the access. That's why the opportunity is lesser. However, indirect methods are more accessible in the market, but they cannot detect the infection at very early stages. So these are just examples of the techniques used for the laboratory-based diagnosis of dengue. So for serology, usually they do ELISA-based detection, enzyme-linked immunosorbent assay for IgM, IgG, and sometimes they use hemagglutination inhibition assays. For nucleic acid testing, you have RT-PCR, sometimes real-time or nested PCR or conventional PCR assays. For virus identification, some laboratories uh, use the plaque reduction neutralization test or PRNT and microneutralization assays. These are tissue culture based virus isolation, and not all hospitals or institutions can afford to put up a tissue culture laboratory. So this is not very cost effective. And eventually, the cost will also affect the access to this technology. There are new technologies in the market today, uh, like Luminex, and there are other neutralization assays based on DC sign or fluorescence and others. The lamp is considered new technology for dengue diagnostics. This table shows the comparison of different molecular assays. For example, for nested PCR, it can detect less than 100 copies of the virus and can detect different. You can do serotyping for all strains, and it is prone to contamination because you need to do two assays, because since it is nested, you will use your uh, uh, two assays. So the first one is to detect if there's dengue, and if there's dengue, you can check if what specific stereotype is present in the patient's sample. And the startup cost is minimal, and but you cannot uh, do a lot. You cannot. It is low throughput since you will use two assays. For NASBA, or this is the one type of isothermal, can also, there are methods that you can determine the serotype, but the startup cost is $25,000, and cannot cannot also automate. There are also real-time PCR assays or kits available that can also serotype, but the startup cost is high. And but you can automate, or you can process more samples. So there are other the cyber green is also real time, and however you can just detect if there's dengue infection but cannot serotype. TMA and LAMP are also like that, isothermal assays, but depending on your design. The biotech answer, there are two types of the biotech specific, the serotype specific uh, and the universal that can only detect one, and the can only tell you that the virus is present in the sample, but will not tell you uh, uh, what serotype if you're using the universal. Actually, the one that is commercially available right now is the universal kit. Because in the field, it will not be cost effective because you will uh, use up more reagents or more tubes for serotype specific. I will discuss later. So, however, the normal lamp, the cyber green based or intercalating dye based detection of the lamp uh, is low throughput. So you cannot process a lot of samples at the same time. We're in the part of the world where dengue burden is high. So the diagnostic challenge, therefore, is how can one access the power of molecular-based assays in the diagnosis of dengue without significantly hurting their pockets, or that they can readily access. So diagnostic kits nowadays need to fulfill this. It should be rapid, accurate, sensitive, specific, and standardized. So what makes a $10 test in the US a $100 test in the Philippines? So it is because of the long value chain. So for example, you are the tech developer, so hindi pa ganun kamahal. However, for example, if the tech developer is from the US, then they need to have a company distributor. And then if you're from, like for example, us from the Philippines, so they will have a separate regional distributor for Asia, and this adds up to the cost of the kit. Then pagdating pa dito sa Philippines, hindi ka basta-basta makakabili because you need a local distributor. So from the local distributor to the technology adapter, or the hospitals, syempre magpapatong pa sila before it gets to the target end user, to the patient. So by the time it gets to the target end user, mahal na siya. That's why there is a decline in the access of that technology. So, konti na lang makaka-access. So, what if we skip some of these steps? So, from the tech developer, you have your technology adapter, and it gets directly to your target end user. So how can this be done? This can be done only through local innovation. Ibig sabihin, tayo ang nag-develop locally, and then the technology adapter is the hospital. Siyempre, hindi na nila kailangan ng distributor, local distributor, because it's locally manufactured. And it can be easily accessed by the target end user. Now I'm gonna tell you about the Biotech and Project and the Labinomic Project. So ano ba to? So basically, the Biotech and Project is the the isothermal technology, not only for dengue but for other diseases as well. So our test case for Biotech and Project was the Biotech and Dengue Aqua Kit, a miniaturized version of the lamp technology that underwent all the stages of technology development and is now ready for clinical application. Actually, it's already commercially available. Such technology allows amplification and easy readouts using the locally designed uh, LED transilluminator and the heater, the isothermal heater. This allows positive targets to light up green when exposed to the blue light and can be used optimally between 0 to 5 days of illness. And it allows identification of the target in an hour with high sensitivity. So it has already underwent laboratory performance testing, pilot clinical testing, full clinical validation, and market research. So the Labinomic project are all diagnostic kits are integrated and miniaturized in an isothermal unit as small as a mug. That's why we call it the Labinomic project, which functions as a multi-infectious disease diagnostic device similar to a portable laboratory. So when you have this Labinamab, you can use it for other diseases, not only for dengue. So the Labinamab project was conceptualized, designed at the Institute of Molecular Biology and Biotechnology of the National Institutes of Health, UP Manila, where I used to work. So this is Dr. Raul Desnura, our lead inventor of the dengue kit. He's also the president and CEO of Manila Health Tech. The technology resulted to two utility models registered under the IPO field, or the Intellectual Property Office of the Philippines. So the Labinamag project consisted not only dengue, but four other technologies like leptospirosis, for typhoid fever, for TB, and schistosomiasis, only under this utility model. So the first utility model is for the kit. The second one is for the Labinamag, the device. So this technology also received a number of awards already since 2016, but the most prestigious one was winning the gold medal award during the 46th International Invention Exhibit in 2018 in Geneva, Switzerland. So before I thoroughly discuss the technology, let me tell you first about the Philippine Technology Act of 2009, or the Republic Act 155 10055. It is an act providing the framework and support system for the ownership, management, use, and commercialization of intellectual property generated from research and development funded by government and other purposes. Now, let me tell you about what is a spin-off company or firm. So this refers to a juridical entity that is an independent business technology taker with a separate legal personality from the GFA or RDA and researcher created through the initiative of the researcher employee who generated the technology. So GFA is government funding agency, like for example, PCHRD or DOST. RDI is a research and development institution like the University of the Philippines. So the Manila Health Tech is a spin-off company. So it is considered as the first ever private research and development company on health biotechnology in the Philippines that is focused on developing and providing affordable diagnostic technologies for communicable and non-communicable diseases. Since the University of the Philippines is not a private entity and therefore it cannot commercialize its own products. To the Tech Transfer Act of 2009, private companies like the Manila Health Tech becomes the technology adapter for biotech and products. So we are actually allowed to create our own company based on the research uh, outputs or the technologies that are generated to the university. So actually, this can be explained by TAPI. There's uh, another office at the Department of Science and Technology who can actually discuss more on this. So these are this is the customized isothermal heater, or we call it the Biotech and Lamp heater. Well, this one is the customized LED trans illuminator where you can view your lamp products. So the Biotech and Dengue Ako kit is lamp-based, or the platform for the technology is liquidated isothermal amplification. Was invented by the Japanese. So they first used it to design DNA targets. So the lamp amplifies DNA with high specificity and efficiency under isothermal conditions. Ibig sabihin, just a single temperature, kaya na niyang i-amplify. Unlike, di ba, sa PCR, kailangan mo ng three temperatures to be able to amplify your target DNA or RNA. So this method employs a DNA polymerase and a set of 4 to 6 specially designed primers that recognize a total of 6 distinct sequences of the target DNA. So the principle is just like PCR. So you have your genomic material, DNA or RNA. So for RNA, you just add reverse transcriptase. So in one reaction, you're already doing the reverse transcription and at the same time amplification of the target. So you amplify, and then the detection is to staging, or some detection methods are like turbidity detection. So you will need additional turbidimeter for that to detect amplification. But for this one, the example here is using an intercalating dye, the cyber green. So what it does, it binds to your DNA, amplified DNA, and then it turns green, a yellow green if it's positive, and remain orange if it's negative. So, ibig sabihin wala siyang ni-bind na DNA. That's why uh, still orange siya. And when you view it under the UV or LED blue light, uh, it will light up or fluoresce. So, so to be able to detect the amplification, you add an intercalating dye like cyber green one. So What it does, like I said, it attaches to your double-stranded DNA and changes the color. You can either detect through naked eye like this one, or sometimes we have had experiences that some of the medical technologies that we trade, they are not able to distinguish the color change, especially if the viral load is low. So pag hindi masyadong nag-change ng color, nahihirapan sila through naked eye. That's why the customized LED illuminator was conceptualized. So under the agarose gel, you can check the positive uh, amplifications by having ladder-like formations. So pag hindi siya nag-ladder-like, like, uh, ganito lang, kunyari, dito lang sa baba, it's negative. So dapat, when you check your amplification, but you don't need to do it, once optimized na siya, you don't need to do the gel. But to confirm your results, you can do this, and you will see ladder-like formations like this. So usually, uh, you see the cyber green dye or the intercalating dye or any, actually there are other intercalating dyes in the market, but the usual one that we use is the cyber green. So since it is an intercalating dye, it does not discriminate from your target DNA to uh, contaminants. So when you open the tube after the reaction, because usually after the reaction you open your tube and add your intercalating dye, um, contaminants may enter the tube if you're not very careful, and that sometimes give you the false positive results. So there is another way of detecting lamp amplification without actually opening the tubes. The method is called quenching of unincorporated amplification signal reporters, or Quasar. We call it Quasar. This method does not reduce the amplification efficiency or sensitivity of the lamp assay. You can still use your normal lamp primers. All you need to do is attach a reporter die to one of your primers and add the quencher uh, that is complementary to your reporter die. So at a defined endpoint, usually 30 to 50 minutes of incubation, the reaction is stopped. So you stop your reaction for 1 to 2 minutes at a higher temperature, like 80 degrees, just like the normal PC. I mean the normal lamp reaction. So and you cool down by removing the reaction tubes to ambient temperature, or you can also put them on ice. And upon cooling, any free primer that has not been incorporated into your amplicon, uh, because for example, there's no target, you don't have a target that uh, 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 no target is detected, the unincorporated uh, primer hybridizes with a quenching probe, resulting in close proximity between the fluorophore and the quencher. So wala kang fluorescence na makikita. So it will be just dark. However, if labeled primer has been incorporated into the amplicon kasi nagamit siya during the reaction, so maghihiwalay sila with the quencher. Hindi siya makuquench, uh, uh, hindi siya it cannot hybridize with your quenching probe and thus remains bright. So you have a bright uh, tube. So you can differentiate already between your negative and your positive without opening the tube. So wala na yung risk of contamination from the environment. Quasar can also be used for multiplexing. Actually, the original developers, Ball et al. In 2016, they were able to multiplex the detection of West Nile virus and chikungunya virus. So if you want to read more about this, this is readily available online, this paper. So if you want to read about the Quasar Lamp. The lamp workflow is actually very simple, from sample preparation to nucleic acid extraction and to amplification. Kasi you just incubate for a few minutes, sometimes 30 minutes, depending on your optimized protocol. And then to the detection, you just use the transilluminator to detect uh, uh, your products. So Lamp is fast, robust. Ibig sabihin kasi same platform, you can use it for other targets depende kung ano yung gusto mo. Actually, not just for infectious diseases, you can use it sa environmental samples or in agriculture to detect diseases in plants, in animals also. So you only need low capital expense, low power, and can work with minimal to no sample pre-treatment. However, yung normal na lamp cannot be easily multiplexed, except for Quasar lamp, you can multiplex. And for other detection, we can have false positives, like if we use cybergreen. So for the biotech and dengue workflow, you just need sample preparation. You, for example, from this EDTA tube, you centrifuge and get the serum or plasma, and then you do extraction using commercially available extraction kits, and then amplification. You incubate in the lamp heater, biotech and lamp heater, and wait for 60 minutes, and then detection using the LED. So you don't need to do anything, or you don't need to add cyber green. Quasar lamp assays are faster and simpler. So compared to your PCR, which requires temperature cycling, the Quasar lamp, or the lamp in general, you just need a single temperature. That's why you can use a normal heater. So the PCR requires 2 primers, and sometimes with a probe if it's real-time PCR. But the lamp requires 4 to 6 primers. And uh, lamp is also typically uh, rapid because it takes uh, less than an hour. You'll have, depending on your optimized protocol, you will already have results. Unlike a normal PCR can take more than an hour to 3 hours. And the yield for Quasar lamp is higher than that of the PCR. But you can also do visual detection in lamp. Unlike in PCR, if you're using the conventional PCR, you need to do agar gel electrophoresis. Or if it's real-time PCR, you need, of course, a real-time PCR machine. And lamp can tolerate sample matrix inhibitors. And you can also multiplex in Quasar lamp, just like the PCR. So LAMP is an ideal amplification technology for point of care or point of need assays. The LAMP actually could fulfill WHO's recommended assured characteristics. So, pasok dito ang LAMP. It is affordable, specific, sensitive, user-friendly, robust and rapid, equipment-free. Well, not really equipment-free. It means just that hindi ganun kamahal yung equipment na gagamitin mo for the assay. And you can deliver it to your end user. So our version of the Biotech and kit underwent already all stages of technology, from prototype to In 2013, we did pilot study and preparation of, well, we did the clinical study, more than 500 samples, and finally, we were able to commercialize it to market it in 2016. And in 2017, uh, DOH already implemented did field implementation. They started with 3 regions, Region 1, 6, and 9. So we did trainings. Uh, and then from 2018 to present, it is already available in the market. So, so these are just examples of the trainings that we did. And finally, uh, the NAAT lamp or nucleic acid amplification test lamp, together with RT-PCR and PRNT or the plaque reduction neutralization test, is already uh, included as a confirmatory test for dengue infections in the Philippines. So through the Biotech and Project, they were able to modify the the confirmatory testing for dengue in the Philippines. So what are the implications of providing and developing local technologies? It's because local technology generates new technologies. So for example, the when we developed the lamp, we were able to conceptualize the heaters and the um, the LED viewer. So it also supports the growth of biotechnology industry in the Philippines. Local scientists also will have more opportunity to hone their skills and need not leave the country. So if we support our local scientists, hindi na magsisialisa na ating mga scientists. They can just uh, do their thing here in the Philippines. And eventually, this will contribute to the macroeconomic growth of the country because we're already developing and manufacturing and selling our own products. So in conclusion, in the period of the reemergence of virus infectious diseases, it is necessary to arm ourselves with the proper tools to combat these invisible agents. Actually, we already have because we have a lot of experts here in the Philippines. So through the ingenuity of Filipino scientists, biotechnology in the country has paved the way for the advancement of molecular diagnostics for communicable and non-communicable diseases. Science, technology, engineering, and mathematics through time has proved its importance in the context of nation building. That, that it has always provided us strong data to back up sound decisions in various fields like our health, economy, environmental protection, and disaster preparedness. So let's not uh, be afraid to take a look. Yeah. So I would like to thank these agencies who helped from the beginning, from conceptualization, from R&D up to commercialization stage of the biotech and dengue and other products, uh, molecular diagnostic products. And of course, I'd like to thank my BRSL family and MTech family. That's all. Thank you.
Thank you very much for that very informative talk, Ms. Santos. I would like to remind our audience, if you have any questions for any of our speakers, please write them on the chat box. Kindly use this format, your name and affiliation, if you would like to identify yourself, then the speaker to whom you would like to address your question, then your question. Then the speakers will try to answer them during the Q&A after the third presentation. Now, our second speaker, Professor Francis Legario, is an assistant professor at the Natural Sciences Department of Iloilo Science and Technology University in Iloilo City. He finished his master's degree in microbiology from the University of the Philippines Visayas in Miagao, Iloilo, followed by postmaster's training in proteomics of cyanobacteria microcystis aeruginosa in Meiji University in Nagoya, Japan. With a scholarship from the British Council DOST Newton PhD Agham, he then went to Scotland for his doctorate, where he is currently a PhD candidate in aquatic pathobiology, specifically fish bacteriology, at the Institute of Aquaculture in the University of Stirling. Today, he will talk about infectious bacterial diseases affecting farm tilapia in the Philippines with his presentation entitled Streptococcosis in Farmed Nile Tilapia in the Philippines, Implications to Food Security and Safety. Let us welcome Professor Francis Legario.
Hello, good morning. Yeah, it's good afternoon there in the Philippines, sorry. Naririnig niyo ba ako?
Yes, sir.
Okay, thank you. So, my presentation is on the streptococcosis in farmed Nile tilapia in the Philippines, implications to food security and safety. So, this is actually a part of my PhD thesis, and that this afternoon will be focusing more on the streptococcosis. And as you can see, this is in the Institute of Aquaculture in the University of Stirling in United Kingdom. So our institute is the only institute in the UK that is dedicated to aquatic sciences, primarily in aquaculture. So let's start with the outline of the lecture. So the lecture would first, we'll describe the genus Streptococcus and the associated gram-positive cocci or bacteria, then their distribution and host. So also have the streptococcosis clinical signs and pathogenesis, and the diagnostic methods of the disease control, zoonosis, and streptococcosis in farmed Nile tilapia in the Philippines. So the genus Streptococcus and related bacteria. So they are gram-positive cocci, so they are encapsulated. Most of them are encapsulated, but there are some strains that have lost their capsule when it depends upon their host. Sometimes evolutionary development will the bacterium loses its capsule. So then they are non-motile, they are oxidase and catalase negative. As you can see it here, the chain of cocci with the white background on them is actually the capsule. So this is a modified Anthony capsule staining. And so they usually morphologically occur in pairs or in chain. You can see the chain morphology, especially when you culture them in liquid medium. So for example, in broth. So when you do your gram staining, you will see the chain morphology of the bacterium. And most of them require blood for their optimal growth. And they can be easily differentiated from Staphylococcus by doing a catalase test. So they are catalase negative and Staphylococci are catalase positive. And if you try to look at in the microscope, the morphologically Streptococci cells are quite smaller than the Staphylococci. And genus Streptococcus formerly included the genus Enterococcus and yeah, your Enterococcus faecium, your Enterococcus faecalis, and your Enterococcus cereus. And we will be talking more on the genus Streptococcus agalactiae and Streptococcus inii. So I'm showing here the phylogeography of the major fish pathogens causing streptococcosis in fish. So these are the pyogenic streptococci. So we have here your S. inii, S. paraibiris, S. agalactiae, S. agalactiae, so this should be Y, Lactococcus garbi, Lactococcus piscium, and Bagococcus salmoninarum. So as you can see here, the Streptococcus inii are global in its distribution. So in America, in some of the Mediterranean countries, here in South Asia, and also in the Far East and in the Southeast. Your Paraubiris is also cosmopolitan in its distribution. The S. agalactiae, primarily in South America and also in the Far East and the Southeast. The Garbiae we have here in the Europe, because this primarily affects the salmon industry, but also they have been found in the coast of Australia and in South Africa. There have been no report yet in the Philippines, but based from our sampling, we have already isolated Lactococcus garbiae from tilapia. So we have detected Lactococcus garbiae and Lactococcus lactis in the Philippines. So Streptococcus agalactiae, this is also known as GBS or Group B Streptococcus based on the Lancefield agglutination test. And it is subclassified into 10 serotypes, serotype 1A, 1B, 2, to 9. And this was first isolated from cows with mastitis. So mastitis is actually the inflammation of the udder of the cow. So this caused a decreased production in milk. So it was named agalactiae because cows with the mastitis have a decreased production of milk. So that's why it's called agalactiae. And primarily beta hemolytic, but some strains are non-hemolytic, especially the fish-adapted Streptococcus agalactiae. There are some strains that do not show hemolysis. And before, there's also called Streptococcus difficile, but later on it was discovered that Streptococcus difficile is actually Streptococcus agalactiae that is non-hemolytic. Streptococcus inii is also a streptococci of importance. It was first isolated from the Amazon dolphin, and it was first isolated from a subcutaneous abscess in 1972, and then it's actually identical with Streptococcus, Shilowi, that caused meningoencephalitis in rainbow trout and tilapia in Israel. So there are two serotypes of Streptococcus inii based on their ability to hydrolyze arginine. So we have serotype 1 and serotype 2. So the distribution and hosts of the Streptococcus agalactiae inii. This figure is from the Aquavac vaccines. As you can see, Streptococcus agalactiae biotype 1. Biotype 1 is also known as serotype 1A and serotype 3. So they are primarily hemolytic. So the biotype 1 is actually quite abundant or endemic here in the Far East and in the Southeast Asia, and here in China, in Vietnam, in Thailand, in Malaysia. But it's not been reported. They have not reported this in the Philippines. So while the biotype 2 or the serotype 1b is quite common here in the South America. We know that South America is also quite very active in aquaculture. So they have found also the Central America and Mexico going downward. So we have the serotype 1b and also serotype on this one, the Streptococcus inii. So it's also found here in South America, in Central America and South America, in Asia. In the Philippines, they only detected biotype 1, no, I mean biotype 2 and S. inii. Okay. So natural infections in fish species. So your S. inii actually infects both freshwater and marine fishes. So more than 40 species of fish are affected by the S. inii. So these fishes with the stars are also affected by S. agalactiae. For example, we have the Nile tilapia, which is the focus of my PhD thesis, and the gilt-heeled sea bream and the rainbow trout. So I think we are also familiar with Asian sea bass. And also, S. agalactiae, more than 36 species are affected by S. agalactiae. So we have seen in the previous slide that it also affects Nile tilapia. So let's go now to the clinical signs and pathogenesis of the streptococcosis. So you can see here, this is a streptococcus infection in Vietnamese red tilapia. This was an image from my supervisor. You can see the hemorrhages, the petechiae in the body and in the fins of and also in the operculum of the fish. So streptococcosis is a septicemic disease of global importance, meaning so this involves almost all the organs in the body where blood comes in contact with, and primarily it involves the brain. So there's neurological involvement in the fish. As you can see here, the fish have a pup eye, the pup eyes, and this indicates actually neurological involvement. That's why the eyes become swollen already. And there is more than one bacterial species involved in streptococcosis. Sometimes you can find both Streptococcus agalactiae and Streptococcus inii in one fish. So, but usually it depends upon the age or the culture age of the fish where the bacteria attacks the organism. It is said to be a multifactorial disease, meaning that the age, the host, the type of species and strain of pathogen can actually affect the outcome of the disease. So the severity of the disease actually. So it depends on usually there are some streptococcus strain that affects early culture period of the fish. Sometimes they also affect only the grow out portion or sometimes on the whole culture cycle of the fish. And it depends also on the species, the strain. So it can cause also acute type of streptococcosis, or it can also cause chronic type of streptococcosis. So I'll show here, this one is one of the clinical signs, so behavioral. So lethargy, loss of appetite, the fish usually do not eat when they are infected with streptococcus, and disorientation. So the spiral swimming pattern here of tilapia, this was taken in our field sampling. So they usually do a spiral swimming. So when this spiral swimming is observed, meaning that usually there is already acute involvement in the brain of the fish. And then you can see the fish, usually the fish goes to the surface because of the, because of the increased ventilation rate. So the fish usually are found on the surface gasping for oxygen. And they are also found usually grouping at the bottom of the aquaria, or also some of them can be found in the side or at the side of the cages of the fish cage. So pumupunta yung fish sa gilid usually. Okay, so what are the clinical external signs? So here we have exophthalmos. So the eyes pops. So this one was, this is also an exophthalmia. The exophthalmos can be bilateral, meaning it involves two of the eyes, or unilateral. Sometimes you can only observe one eye that is swollen. Then we have corneal opacity. As you can see here, there is a corneal opacity of the eye and petechial hemorrhages in the body of the fish. And also you can see redness in the body. Then we can see abdominal swelling, anal inflammation, discoloration, skin lesion, or anorexia. But not all of these clinical signs can be found in the fish. Sometimes you only find exophthalmos, sometimes you only find abdominal swelling, sometimes you only find petechial hemorrhages. But these are some of the clinical signs to look for in moribund or sick fish. In internal clinical signs, so the fish usually have ascites. So you can see the yellow fluid oozing from the abdominal cavity. And they may present splenomegaly, hepatomegaly, and pale liver. We can see here is the enlarged kidney. Sometimes there are also internal hemorrhage, gill pallor, and meaning gill pallor. So there is pale gills, no? So the gills becomes pale, pinkish, not red, and intracranial edema. So you can see that the brain usually becomes swollen. So let's go to the pathogenesis of streptococcosis. So the pathogenesis is not yet fully understood, but we know that the pathogen enters into the bloodstream. And the pathogen utilizes macrophages to cross blood-brain barrier. So we call this one as the Trojan effect, meaning that the pathogen, when they are phagocytized by the macrophage, they produce some virulence factor that will not allow the macrophage to kill them inside. So they become dormant inside the macrophage, and the macrophage being in circulation, it goes to the different organs of the body, to the spleen, to the kidney, to the liver, and also it crosses the blood-brain barrier because the bacterium itself cannot cross the blood-brain barrier, so it needs a vehicle. So it uses the macrophage. So that's why we call that one as the Trojan effect. So it enters into the brain because the streptococci have a particular affinity to the brain. So they like to go to the brain. And they may disseminate as free bacteria in the system. So they go to the heart, to the intestine. So when that particular thing happens, we call that already as septicemia. So transmission is commonly horizontal, but has also been shown to be vertical. So there is also a paper that reports a vertical transmission of the streptococcosis in Nile tilapia. And in terms of tissue tropism, so as I have said, the streptococci have the affinity to brain of the fish. And temperature is an important predisposing factor in disease development because increase in temperature usually is observed. When there is an increase of temperature, there would be what we call a outbreaks of streptococcosis because the temperature will actually cause increased expression of the virulence genes of the microorganism. So, diagnostic method. So, the streptococci, usually we sample from the kidney, from spleen, and from the brain of the fish. Usually we do not actually get from the brain. Kidney and spleen are already enough for sampling for bacterium. But when we suspect that the fish is actually having streptococcosis, so we also get the brain, but usually kidney and spleen are enough, and the brain is only used for histopathology. So we use general purpose media, for example, TSA or BHIA, and the colonies of the streptococci are usually small, white, translucent colonies. So normally, they grow after 48 hours to 72 hours at 28 degrees Celsius. So they're kind of quite slow growing in TSA or in BHIA. So usually we do add blood. So, but you can also use the selective medium, the Edwards medium. So together with antibiotic, where the colonies of the bacterium appears small blue to bluish white colonies. So this is actually these are streptococci, which is in your Edwards medium. So conventional identification. So yeah, they are gram-positive cocci in pairs and chains. They are oxidase and catalase negative. They are non-motile. They have no reaction in your oxidation fermentation test with glucose, hemolytic activity, but not every one of them, or not all streptococci, especially the Streptococcus agalactiae, produces hemolysis. So the hemolysis is quite not that big as compared with other bacteria. For example, your Aeromonas, you can see very big zones of hemolysis. The hemolysis in streptococci are quite small, and sometimes they are obscured by the production of green pigment from the red blood cells. So API test, so usually there's little reaction using API 20E. So we use API 20 strep, but this is not a reliable ID because of the varied biochemical reactions of the different strains, especially those that are fish-adapted strains of streptococci. Hemolysis, so as you can see it here, as I have said, the zone of hemolysis in streptococci are very small. So you usually observe hemolysis after maybe 48 hours to 72 hours. Usually, so we have alpha, beta, or gamma hemolysis. So the alpha group, which includes the Streptococcus pneumoniae, so actually actually that is not actually a total breakdown of the red blood cells. And the beta hemolytic group, so they are more destructive of red blood cells, and sometimes they are called hemolytic streptococci. So that's for the hemolysis. And for Lancefield antigen or test, so we have different groupings here. So we have group A, B, C, D, E, F, and G. So group A are usually pathogenic to humans, and group B and group D, these are the fish isolates that are actually pathogenic to fish and yeah, pathogenic to fish. So group B, so this is the Streptococcus agalactiae, and it is the only Streptococcus species that has this Lancefield cell wall antigen B. So when we speak of group B Streptococcus, it is synonymous with Streptococcus agalactiae. So we have here an example of a commercial kit, which we call the slide strepto kit. you can see here agglutination in the group B. Actually, this slide streptokit is not available commercially, currently not available commercially. I think they have already stopped production of this because in terms of accuracy, it will be dependent upon the technician to observe agglutination, and it's time sensitive. So instead of doing slide agglutination tests, we can do PCR, which is more accurate than this agglutination test. One of a simple test to differentiate Streptococcus agalactiae from Streptococcus inii is starch hydrolysis. You can see here, this one is Streptococcus inii, which is positive for starch hydrolysis, while here the Streptococcus agalactiae is negative for starch hydrolysis. But it doesn't mean that S. inii is the only Streptococcus that is positive for starch hydrolysis. There are also other streptococci that are able to hydrolyze starch. But if you think that your isolate is either agalactiae or is inii, so this can add value to identification from your PCR maybe to do a starch hydrolysis, just to confirm. So we also have molecular identification test. So yeah, 16S as the gold standard. Then we have species-specific PCR. So we have here different primers. These are useful, but and useful but limited. So because of the high investment and the high technical skills involved, but it is high sensitivity. And we also use molecular serotyping to serotype the different streptococci. So this one is a species-specific duplex PCR for the identification of S. agalactiae and S. inii. Actually, we developed this one at the University of Stirling. So here, we use the lactate oxidase gene of the Streptococcus inii, and then we also have the 16 to 23S interspacer region of the S. agalactiae. So you can see it here that this one is the positive control for S. inii, and this one is the positive control for our S. agalactiae, and these are our samples 4 to 10 and 11, and this is the negative control, I think. So the duplex PCR, so it will allow you to differentiate, differentially identify S. agalactiae from S. inii. So these are for the S. inii, it's around 870 something, and for the S. agalactiae, it's around 260 something. So this is very helpful in the identification of correct identification of the streptococci. In molecular serotype, we usually use multiplex PCR. However, for piscine or fish S. agalactiae, they usually belong to serotypes 1A, 1B, and 3. So if you are doing molecular serotyping for, think a fish isolate, you don't need to use all of the primers to amplify other serotypes. You can just use 1, 2, 3. But if after your molecular serotyping, there is an isolate that is not either of this serotype, then you can do the rest of the serotyping. So there are current issues with diagnosis in streptococcosis. So there, because of the range of organisms, cause similar pathological signs, meaning that streptococcosis is not pathognomonic. So meaning that the disease, as the same with other bacterial infections in fish, they present the same clinical signs, just like exophthalmia, ascites, dropsy. And the severity of the clinical signs is varied. So it depends on the age of the tilapia, or on the sex, on the species, on the environment, or the systems. So traditional bacterial identification methods are unreliable, and molecular tools are improving. So used in combination with traditional bacterial identification methods are molecular tools. It's tricky to differentiate closely related species. So if you want to really to differentiate them, you can use pulse field gel electrophoresis or multi-leukosequence typing, but it's really quite expensive. And yeah, it requires investment and high expertise. We are also actually doing MALDI-TOF, matrix absorption laser deionization, some like mass spectrophotometry. But MALDI-TOF is only a value when you have a pure culture. You cannot use MALDI-TOF from clinical samples. So the protein spectra, the spectra is also dependent on the deposited spectra. So this is actually nice technology for ID, but yeah, as I have said, it requires pure culture, and the protein spectra of the bacteria is dependent also upon the temperature of the incubation temperature, the culture medium, and the extraction methods. So you can also have viable recovery using chromogenic agar, but chromogenic agar is very expensive. As you can see it here, you can easily differentiate S. agalactiae because of this is a purple colony. So if you have a clinical samples, so you can easily isolate them by the difference in their color of their colony. But you know, yeah, as I have said, chromogenic agar is quite expensive. So in terms of zoonosis, S-agalactiae and S-NEA are of concern because of their ability to infect humans. So they do not only infect fish, but they can also infect humans. So they are implicated in human infections, especially those that are immune compromised. S-NEA is primarily due to food handling, which can develop into cellulitis, disseminate into the bloodstream, and cause endocarditis. So S. agalactiae sequence type 2A3 infections, which is foodborne outbreak, which actually happened in 2015 in Singapore. The patients developed meningoencephalitis, quite scary, septic arthritis, and spinal infection. So this is one hypervirulent clone that was both isolated from deceased tilapia and from humans. So when they did their study, they were able to point out that it was due to consumption of tilapia. Well, raw, when you do not properly cook the fish, then you may be infected with the bacterium, especially when the fish do not have the clinical signs. So we call that one as subclinical infections. The fish have subclinical infection. It doesn't show on the outside external signs of infection, and then you eat the fish raw. Then it has the ability to cause zoonosis. So it is actually, these two bacteria are actually a food safety issue in fish. So let's go now to my study. Actually, the study here we conducted in the Philippines. So we know that tilapia is primarily done in freshwater lakes in the Philippines. So we have Taal Lake, Laguna Lake, and other freshwater lakes in the Philippines. So nakita natin most of the tilapia in Manila, the supply comes from Taal. Hindi ko sinasabi na lahat ng tilapia sa Taal Lake infected ng streptococcus. So we did our sampling also in Pampanga, in Nueva Ecija, in Bacolod, in Iloilo, in Mindanao. So the volume of production of tilapia over the years is quite the same, if not decreasing. although meron tayong mga improved tilapia strains, and we have already expanded into the brackish water, the question is, why is it that hindi pa rin tumataas ang production? Although, yeah, we do not export tilapia because the demand locally of tilapia in the Philippines is quite high, especially in the National Capital Region. At mas mataas yung price ng tilapia in the local market than in the international market, because South America also produces a lot of tilapia, Vietnam, Bangladesh, India. So before, sabi, akala natin tilapia is a hardy fish, but we now recognize that it is susceptible to diseases, just like your tilapia lake virus and bacterial diseases. So there is mortality outbreaks annually. Actually, sa Taal Lake, parang common na nga yung streptococcosis, they call that one as hibay. It is an annual occurrence na. So what happens is that during our interviews in the farms, the survival of the tilapia is actually 10 to 20%. It's quite very low, yung survival nila. So what they do is they try to stock high the tilapia. Mataas yung stocking density nila. Instead of 50,000 in a 10 by 10 meter cage, ginagawa nilang 300,000, parang survival of the fittest ang nangyayari. So because of this, because in 300,000, only 10 to 20% will survive. Pero the negative impact of this is that because malaki yung stocking density, ang culture period nag-extend. Instead of having 2 production cycle in a year, nagiging 1 production cycle na lang yung culture ng tilapia in Taal Lake. So that's one or maybe of the reason why hindi nag-i-increase yung production. So umi-increase yung population natin, pero yung ating food production is decreasing or still the same over a 10-year period. No, malaki, I mean, it's quite a long time already that our production is not increasing, but our population is increasing. So the demand for food is actually increasing, but our production is the same or if not decreasing. So we did our field work in here. So I have said Nueva Ecija, Pampanga, and we have here in Taal Lake, in Laguna Lake. Yeah, I think this, yeah, Laguna Lake. In Laguna also, we have hatcheries here. We have grow out. We have also in Panay, in Negros, and in Davao. This is on marine tilapia. So as you can see here, ang dami-dami na talagang cages sa Taal Lake. That could be one of the factor on the continued outbreak of the disease, kasi yung holding capacity, yung water quality ng lake ng Taal is hindi na maganda. Stress na ang fish, madali silang kapitan ng infection. So you can see here, yeah, we have bilateral ectophthalmia, we have anal swelling and hemorrhage in the brain. So this is our isolation of the bacteria. Then we have conventional identification methods. So we did all of the conventional identification method. We also did antibiotic susceptibility testing for the bacteria. So for the 6 here antibiotic that is approved in global aquaculture, and then also we did additional antibiotics just like chlorphenicol, vancomycin, ampicillin. It's good that our Streptococcus agalactiae ini are only resistant to oxacillin. No, not oxacillin. I mean oxolinic acid. But there are some strains of S. agalactiae that are resistant already to SXT, especially the serotype 1b. So molecular identification. So we did our duplex PCR, as you can see here. So these are our positive and negative control. So this is the S. inii, these are our S. agalactiae isolates. Then after we did our duplex PCR, we confirmed some samples by 16 srRNA PCR just in order to confirm the specificity and the accuracy of our duplex PCR. This is molecular serotyping using the CPS, the capsular gene. So this is our serotype 1b, that's our positive control. So you can see here the 3 bonds, 688 BP, 621, and 272. Serotype 1a will only have 2 bonds. So you have 688 and 272, while the serotype 1b will have the 3 bonds. So we also detected virulence genes. So the virulence genes for SNA, all of the SNA isolates have those virulence genes. So ibig sabihin, sa isang clonal lang talaga, isang clone lang talaga yung SNA in the Philippines. So all of them have this virulence genes. This one, SAG-A is actually an invasine, so it is a cytolysine, so it allows invasion. SIM-A and PDI are adhesine, and CPSD, PGM, and SCPI are immune avicine. For S-agalactiae, all S serotype 1A have all of the genes except for serotype 1B. Sa serotype 1B, wala silang SILE gene. So, pero ang lahat ng serotype 1A, mayroon silang lahat ng genes at SILE. So ano itong SILE? The SILE gene is the structural gene involved in hemolysin. So as you can see, this one is hemolytic. These are non-hemolytic. You can see the colonies here. Wala silang zones of hemolysis. So the SILE is actually very important in the establishment of streptococcal infection. So in terms of histopathology, you can see here gram stain sections of the brain of the tilapia. So this one A is with meningoencephalitis due to Streptococcus inii. You can see here the macrophages filled with the bacterium. This one is also seen in serotype 1b, while this one is a meningitis seen in serotype 1a. You can see the bacteria that are within the brain capillaries, but you cannot see the presence of macrophages filled with the streptococci. So, ano ito? Ang implication is that Streptococcus inii and serotype 1b are mostly involved in chronic infection, while serotype 1a is in acute infection. So, overall findings of the study. So S-inii and S-agalactiae are the 2 main causative agents of streptococcosis and farmed Nile tilapia in the Philippines. So S-agalactiae is widespread, while S-inii is localized. And there were 2 serotypes of S-agalactiae that were detected. So we detected serotype 1a and serotype 1b. Between the 2 serotypes, serotype 1a is more dominant than serotype 1b. And ang Aquavac, hindi nila na-detect ang serotype 1a. So this is the first paper actually that detected the serotype 1a in the Philippines, and this is the more dominant serotype. While the serotype 1b is said to be the less dominant type, but this is implicated more in chronic infection as these bacteria were recovered more from already quite mature tilapia fish. Ang serotype 1a, pagpunta namin doon is there is already outbreak, and we isolated this more from 2 to 3 months old tilapia. So this is the first report of Paisine S-agalactiae small colony variants. Ano itong small colony variants? This small colony variants are a variant form of bacteria that have that are metabolically impaired. So ang small colony variant ng S. agalactiae are non-hemolytic also because from the studies, pag hemolytic yung, overly hemolytic yung bacteria, for example, yung S. agalactiae, madali silang i-destroy ng macrophage. But if they are not hemolytic, they can establish themselves more in the macrophage. As you can see in the meningoencephalitis of the serotype 1b and S. inii, less hemolytic, mas efficient sila in Trojan horse effect. Okay, so there are subtle differences in the histopathology in S. anii and S. agalactiae, and also on serotype 1a and serotype 1b. So overall, we have found that streptococcosis is one of the main factors, that is involved, that is implicated in the decrease of, or in the decreased production of farmed Nile tilapia in the Philippines. So this study is published in the Journal of Fish Diseases. So we just have published this one. The paper came out just this September. So if you would like to read more on this study, you can easily access the paper because this paper is open access. So we have here isolation and molecular characterization of streptococcal species recovered from clinical infections in farmed Nile tilapia in the Philippines. So yeah, with my co-authors here. So you can access this in Journal of Fish Diseases. So wala kayong babayaran because it's open access. So that's all. Thank you. And yeah, I'd like to acknowledge the British Council, the Newton Agram Program, the Iloilo Science and Technology, the DOST, CIFDEC, and the Fisheries Biotechnology Center, TUCAS, and to the technical staff there for their great help during sampling. I know some of them are also attending here the webinar. And also to my two friends, Justin, who's doing his PhD also in environmental science in University of Edinburgh, and Romel, who's doing also his PhD in Zika virus at the University of Glasgow. So tatlo kami in Scotland. So thank you.
Okay, thank you very much, Professor Legaria, for another fascinating lecture. Again, if you have any questions for our speakers, please submit them using the chat box. And please stay on until the end for the open forum and the link to the evaluation form. Now, our last speaker, Dr. Marie-Christine Obusan, graduated with a PhD in Environmental Science from the Institute of Environmental Science and Meteorology, University of the Philippines, Diliman. She is currently an assistant professor at the Institute of Biology, also in UP Diliman. Dr. Obusan is also the principal investigator of the Microbial Ecology of Terrestrial and Aquatic Systems Laboratory, or METAS Aquatic Lab. Her work is on microbial ecology of aquatic systems with focus on sentinel species and habitats. Her current research on cetaceans of the Philippines include the investigation of emerging leptospirosis and toxoplasmosis, antimicrobial resistance, and molecular detection and immunohistopathological confirmation of coccidian parasites. Her other research interests include microbial communities as sentinels of emerging environmental pollutants in water resources. Her talk today will be on Cetacean Health and Stranding Response Program, a One Health initiative in the Philippines. Please welcome.
Please welcome Dr. Marie Upusan for ma'am.
Hi, good afternoon.
Good afternoon, ma'am.
Let me share my screen. Okay, can you see my screen now?
Yes, ma'am.
Okay. Once again, good afternoon. Thank you for the introduction. Magandang hapon sa lahat. I would like to thank the BRSL for this opportunity for me to share our research work involving marine mammals in the Philippines. So for this afternoon, I will be presenting our cetacean health and stranding response program, which is our One Health initiative. So let me start by introducing marine mammals. So like us, they are mammals exhibiting the following traits. They maintain internal body temperature, they have hair or fur, they breathe air through their lungs, they bear live young, they nurse their young with milk produced by mammary glands. And this particular group is not a formal taxonomic group. We just group them and call them as marine mammals because they have developed common adaptations for aquatic lifestyle. But please take note that not all marine mammals are inhabiting marine waters. As Sir Francis Legario mentioned in his talk, a particular Streptococcus acti was obtained from an Amazon River dolphin. So we have marine mammals inhabiting freshwater ecosystems. So actually, mas appropriate na tawagin silang aquatic mammals. But nevertheless, we are used to calling them as marine mammals. So as a group, this is represented by three orders. We have order Carnivora, such as otters, seals, and polar bears. Then we have order Sirenia, such as the dugong, for example. So you know the manatees and sea cows. And then we have order Cetacea. So the cetaceans are represented by the whales, dolphins, and porpoises. So this group is actually divided into two. We have the odontocetes, or those animals provided with teeth for the consumption of their prey. And then we have the mysticetes, or those animals provided with baleen plates for filter feeding. So I'm showing here the general perspective of the anatomical parts of cetaceans. But I just would like to emphasize here the presence of the blowhole, which is comparable to our nose. So this is where they breathe, okay? And it's a small hole on top of their head. So I'm emphasizing blowhole here because there are some cases in which responders during stranding events accidentally put water into the blowhole, and that causes the animal to drown. And I have here the updated list of marine mammal species in the Philippines. So we are a proud host to 1/4 of the world's marine mammal diversity. So we have a total of marine mammal species, a total of 31 marine mammal species in the Philippines. And out of these 31, we have 29 cetaceans. So this is this is a diverse assemblage which deserves conservation and management, and also scientific attention. And it is only in the recent years that we are studying this particular group of animals for their microbiota. So out of this species, we have 6 species which are actually listed under vulnerable and threatened national and global list. So this deserves concerted effort because these animals are facing the threats of human activities. So I would like to share what we're doing right now. So from 2017, we have been isolating bacteria from cetaceans, and we're detecting their antibiotic resistance profiles, their virulence factors, and their antibiotic resistance genes. We are also investigating our cetaceans for the presence of leptospira, the causative agent of leptospirosis, and Toxoplasma gondii, the causative agent of toxoplasmosis. Also, we have started our histopathological assessment to detect coccidian protozoan parasites and to associate and corroborate our findings in the case of suspected bacterial infections. So why are we doing this? This is the paradigm of why we are investigating cetaceans. We are using them as sentinels, which means animals that can indicate emerging pathogens and diseases in our case. So this is under the perspective of One Health, which aims in general to know the interconnections among cetaceans, marine environment, and humans. So this is not the first time that I have explained the perspective of why I am studying cetaceans in the Philippines. So in all my talks, I always make it a point to explain why we are using cetaceans as sentinels. So our biggest challenge is actually the collection of biological specimens. These animals are relatively big. Nasa gitna sila ng dagat. Most of our species are offshore species or pelagic species. We have many deep diving species. And for us, for example, to isolate bacteria, we need to obtain swab samples of their cavities. We need to obtain blood sample, for example, for us to detect the presence of Toxoplasma gondii gene. So that's very difficult because we need a lot of research money for us to have a dedicated vessel, for instance, or a dedicated team that can sample their populations in the wild. And they're relatively big. Medyo malalaki sila as compared to other animals which we can study or investigate in the lab. So what we're doing is that we are obtaining our biological samples or specimens from the individuals that get stranded in our waters. So on standby kami nationwide for cetaceans that will be stranded. So how do we define a stranded cetacean? It is any dead cetacean or any live cetacean that is cast ashore or unable to return to its natural habitat. For example, I have here a live strander, which is identified as a pygmy sperm whale, which was found and responded in Batangas last April 10, 2019. And on the left photo, you can see here a stranded pantropical spotted dolphin. Patay na siya na na-strand. So this was responded in Quezon last April 9, 2019. So in terms of collecting biological specimens, a live animal or a dead animal are equally important because in the case of dead animal, we can perform necropsy, we can conduct gross morphological observation, and we can collect different tissues. In terms of live stranded cetaceans, we can obtain swab samples, we can extract blood, and we can also observe their behavior during the stranding response and rehabilitation or management. Maybe you've heard about recent mass strandings of cetaceans this 2020. So for example, we had the recent short fin pilot whale stranding that occurred in Sri Lanka last November 3. Then we have the melon-headed whale stranding in Catanduanes last October 7. So both of these mass strandings involved approximately 100 individuals. So strandings can be single or mass. So when you say single, isang hayop lang yung na-strand na tagpuan sa beach. And in the case of mother and calf, we also consider that as a single stranding kasi hindi naman nagsusolo yung baby. Parati niyang kasama yung kanyang mother. So mass strandings involve more than one individual except in the case of mother and calf. So imagine we have here the stranding of approximately 100 individuals. The stranding network actually responded to the melon-headed whale stranding in Catanduanes. So what are the factors causing marine mammal stranding events? Hindi lang naman kami naghihintay para may ma-strand yung dolphin or whale. Hindi rin kami natutuwa kapag na-strand sila. Because you might think, we need to obtain samples. So baka natutuwa kami when they get stranded in our waters. So actually, our primary aim is to help the animal in need. So the research team in collaboration with the Stranding Network first provides aid to this stranded animal and evaluate the animal. So we assign particular codes, preservation codes, and animal disposition codes. So when we're sure that the animal is already stable, that's the time that we can perform biological sample collection. But how do we evaluate marine mammal strandings? What are the factors causing them to strand? It's actually difficult to arrive at a definitive conclusion. And most of the time, it's a combination of different factors. So some of the factors that I can mention are fisheries interaction, extreme weather events such as earthquake or typhoon, predator interaction. You have here a shark attack, as you can see, sonar activities and acoustic trauma, such as acoustic trauma from dynamite blast, for example. And of course, infections and diseases. So my research team is trying to know the association of those stranding events to infections and diseases, specifically the emerging diseases. So when we say emerging diseases, those are diseases that were previously unknown to affect our cetaceans here and worldwide and elsewhere. And they are now observed in different populations and different species in various geographical regions. So I would like to present here the updated data about our cetacean stranders. So as of 2019, we have this collated data by Aragones and Lagui. So this data were documented from different administrative regions of the Philippines from 2014 to 2018. We have a yearly average of 105 cetaceans that get stranded in our waters. So ang problema na lang namin ay kung paano pupuntahan sa iba't ibang parte ng Pilipinas itong mga na-strand natin na cetaceans. So far, we have the so-called regional hotspots, such as you can see here in the photo, you have the stars, the red stars there. So the regional hotspots are regions 1, 2, 3, 5, and 7. So most of the frequencies are being recorded from these administrative regions. So imagine the wealth of samples that we can get if we have a yearly average of 100 cetaceans. So actually, in the case of microbiological research, it's very crucial that we obtain samples and bring them to the lab within a particular period of time for us to, isolate bacteria, process the serum, process the blood. So through the years, we have optimized our specimen collection, and we're glad to have established our collaboration with the Philippine Marine Mammal Stranding Network and the Bureau of Fisheries and Aquatic Resources. So the BFAR has their provincial and regional offices, which also coordinate with PMMSN and with our lab with regard to the sending of samples. So members of the network respond to cetaceans during their stranding events. And then we have microbiologists and veterinarians who were trained to collect samples. And then these specimens are transported to UP, and then we store them and we process them for different research applications. So to facilitate that nationwide system, we have distributed sampling kits. So we have assembled kits, reagents, materials that our veterinarians and fish officers can use in the field. We have also developed the manual, which is a work in progress. We keep on revising the manual based on the context and logistics of responding to cetaceans for the purpose of obtaining samples for our microbiological research efforts. And through the years, PMMSN also has been conducting medical management training and microbiological sample collection training for veterinarians. So the last training that we had was last January 2020. So through these efforts, we are slowly building our cetacean repository, wherein we have frozen tissue samples, we have buffered tissue samples, we have DNA samples, and then we have now our bacterial culture collection. So this is housed primarily in Marine Mammal Research and Stranding Laboratory at the Institute of Environmental Science and Meteorology in UP Diliman, and of course in our lab at the Institute of Biology in UP Diliman also. So this is the hardworking team of the Microbial Ecology of Terrestrial and Aquatic Systems Laboratory, specifically the Metas Aquatic Division. So these are my students and research associates processing the samples from cetaceans and performing different downstream applications. And then we also have field trips and survey expeditions. So one of the research projects that we have is the detection of Leptospira in cetacean. So most of you, I think, are familiar with Leptospira species, gram-negative bacterium that is the causative agent of leptospirosis. So for us to detect them in cetacean tissues, we use several assays. For example, we use the enzyme-linked immunosorbent assay for antibody detection. We also culture the different samples. So for this particular project, we used blood, kidney, and urine samples. And then we also use molecular detection techniques to confirm the identity of the isolates. And we are also genotyping them using the multi-locus sequence typing analysis. So aside from this, we are also performing histopathological assessment because we want more evidence of actual infection in the animal. So for this, we need actual tissue samples from necropsy animals, but not all animals end up being necropsied. So at least we have blood and urine samples for us to culture the bacteria. And then another project is the detection of Toxoplasma gondii in cetaceans. Like the Leptospira, the Toxoplasma gondii is considered an emerging pathogen. So both of these microorganisms are predominantly terrestrial, and due to anthropogenic activities, they now find their way into the marine ecosystems, and now we are detecting them in marine mammals. So in the case of Toxoplasma gondii, we are using serological assay for IgG detection. We also detect the RE gene of this microorganism, and for their genotyping, we use multi-locus nested PCR restriction fragment-led polymorphism analysis. And also, we are performing histopathological assessments to detect the cyst. And then in terms of the bacteria that we isolate from cetaceans, of course, we perform phenotypic and molecular identification techniques. So for phenotypic identification, we use Vitek. And for molecular identification, we have the usual 16S. And then we determine their antibiotic resistance profiles. And also we detect the antibiotic resistance genes and virulence factors. For histopathological assessment, so we do this in close coordination with the veterinarians that were trained for medical management of cetaceans. So this usually starts with the gross examination of organs, collection of tissue samples, and then the usual sectioning, processing, and fixation of tissues. Then we use different stains depending on our target lesions and target parasites. And then we perform microscopy in the lab, and based on our microscopic observation, we corroborate our findings with the necropsy reports that are forwarded to us. One of the efforts under histopathological assessment is the detection of coccidian protozoan cysts. So specifically, I have already mentioned Toxoplasma gondii, but in addition to that protozoan parasite, we are also detecting Sarcocystis species. So these coccidian cysts were first reported among free-ranging harbor seals and sea otters. And it is only lately that they are being reported to have been detected in cetaceans. So for this, we use lungs, liver, kidney, and brain samples. I forgot to mention that in the case of toxoplasma, we use also the blood and brain samples and then the cardiac skeletal muscle samples. We also started optimizing this method for confirming the presence of gram-positive and gram-negative bacteria in tissue. So we are optimizing a modified gram staining procedure which will enable us to quickly detect the presence of this bacteria in cetaceans suspected to have pneumonia during rehabilitation. So we're still optimizing this, but this is a rather quick method for us to evaluate if there is an infection. So this is the synopsis of our microbiological findings. So you can see here the year of report or research effort, whether the findings have been published or not, and then the number of sample citation stranders, the year of sample collection, the percentage of total number of stranders for that particular sampling period, and the pathogen prevalence, depending on our target species. So some of the findings have been published already. Then we have submitted papers recently. Then we have this ongoing efforts with incomplete data. So in general, you can see here the percentage prevalence. And you can notice here that when we compare the percentage of total number stranders for a particular period and the number of citation stranders that we are actually, investigating based on the samples that we're able to retrieve in the lab, we are not maximizing the available um, citation stranders in the country. For example, the highest pool of samples that we got is 40, which is only 17% of total number of stranders nationwide. So if you want to get a copy of the different papers that we have published, you can email me. I will show my email address later. So what we know so far is that we have Leptospira species in our cetaceans. So far, we have detected this bacterial group in 7 cetacean species: spinner dolphin, Fraser dolphin, melon-headed whale, pantropical spotted dolphin, short-finned pilot whale, and pygmy sperm whale. So our report is only the second report in terms of Leptospira detection in cetaceans. So this particular bacterial group was first reported in southern right whales in Argentina. And most of reports on leptospirosis in marine mammals involve sirenians, sea cows, manatees. So in terms of antibiotic resistance, you can see here that in general, we are detecting a high percentage of multi-drug resistance. So at least 50% in the case of data sets that we have completed. So this 40% here is only our partial data. So you can see here the different bacteria that we identified and the different antibiotic classes tested. So please take note that these are not clinical isolates. So we consider bacteria as clinical isolates in the case of cetaceans, if the stranded individual is already showing clinical symptoms under rehabilitation and medical management. So this is actually a baseline data that we forward to our veterinarians for them to be informed on what antibiotics may work, okay, on patients that they are managing during rehabilitation process. So as you can see here, we have tested a lot of antibiotics, and it's actually this one is actually very significant because of the high multi-drug resistance. So this is a representative data of our detection of antibiotic resistance genes. You can see here the enumeration of the different genes that we have detected based on the bacteria ID and their percentage occurrence. We're also detecting virulence factors. So we have started optimizing our methods for this. So I'm presenting here some examples of virulence genes that we are detecting. Okay, and the choice of these virulence genes depend on the bacterial group. So we are also updating our list of cetacean species that we are sampling for our pathogen surveillance. So as of 2020, for instance, for the detection of Leptospira, Toxoplasma, and Coccidiensis, we have this 14 cetacean species. And most of the species that we have sampled are Codia breviceps, or the pygmy sperm whale, and Stenella attenuata, or the pantropical spotted dolphin. In terms of cetacean species sampled for antimicrobial resistance as of 2020, so far we have sampled 11 cetacean species, and most of these are Laganodelphis hussae, or the Fraser's dolphin, and Globicephala macrorhynchus. So I would like to end by sharing my insights and my thoughts on cetacean research in the Philippines based on our experience in the lab. So we've learned that we can always plan, propose, optimize methods, and anticipate for the collection of biological specimen from stranding events nationwide. But ultimately, the performance of the methods that we have optimized in the lab will always depend on the quality of samples that we retrieve from the different stranding sites. So there were times when we have wasted our serological kits, for instance, because we were not able to obtain qualified serum samples for our method in the lab. Also, we have wasted many swab samples because we were not able to retrieve the samples in time for bacterial isolation. So we do hope that we can develop and strengthen our capacity to use non-culture-based techniques in studying the microbiota of marine mammals in the Philippines. But for us to do this, we need more microbiologists who would be interested in joining our team or conducting studies involving our marine mammals in the Philippines. And also, we would like to encourage our participants here to be involved in our stranding network. So the Philippine Marine Mammal Stranding Network actually provides training workshops every year for those who want to join the team. So they will learn the basics of responding to a stranded individual, and advanced training would be uh, for the collection of microbiological specimens. So I'm going to show a video clip later But before that, I would like to thank you all for your attention this afternoon. And this is my email ad if you want copy of any publication that we have. And you can also visit our lab website and our Facebook group. And I would like also to thank the members of our team in this initiative. So we have here the list of the names and the different students who have contributed to our wealth of findings. And of course, the different laboratories and agencies and stakeholders collaborating with us in this citation response and health surveillance program. Thank you.
Thank you very much, Dr. Obusan, for that very enlightening and inspiring talk. So I will now, yes, ma'am.
Ma'am, Jude will play the video.
Okay po.
Thank you. Thank you.
Thank you very much, Dr. Busan. So we will now open the open forum. So I'll ask the first question. This is for Ms. Joyanne Santos from Leonel Lumogdang. What dengue serotype does the technology detect? Does it detect other serotypes? And what molecular markers does it detect and signal into our lamp?
Hi, actually, the Biotech and Dengue Aqua Kit that is uh, commercially available right now can detect all four serotypes, but you cannot differentiate which serotype kasi that will be more expensive. In the clinical setting naman kasi, ang importante, ma-detect lang yung presence ng virus. You don't need to know the serotype unless surveillance yung studies na gagawin.
And then, so what text signals into our lamp?
Sorry, the Biotech and Dengue detects the NS5, non-structural protein 5 of the dengue virus. It is a conserved region. Did I answer your question?
Yes, ma'am. Yes, ma'am, Jab. Thank you. Thank you for that answer. Let us move on to the next question from Mampai. Are there any other non-microbial endosymbiont associated with Streptococcus? This question is for Sir Ligario. Sir, your mic is muted.
Paki-repeat nga ng question eh. Yes. Ano yung question?
Sir, the question is, are there any other known microbial endosymbiont associated with Streptococcus?
Based from our sampling in the Philippines, we have isolated, aside from Streptococcus agalacti and INEA, we have Lactococcus garvi, Lactococcus lactis, Enterococcus hirae, and some other enterococci. But right now, we have also Aeromonas hydrophila, Aeromonas veroni, Aeromonas dickensis, and Aeromonas allosaccharophila. But the presentation only focuses on streptococci, but we are doing other tests and research on the other bacteria that we were able to isolate from the deceased tilapia in the Philippines.
Okay, sir, thank you. We have another question again for you, sir, from Aaron. Are there any remedies or strategies to help eliminate or decrease Streptococcus infection in tilapia farms?
Actually, we're trying to, the gist of the study is that later on we're trying to develop a geographically distinct vaccine for streptococcosis, but I don't know the take up of the vaccine would be, if it's acceptable to our farmers, because we know vaccine is quite expensive and tilapia is very cheap. And economically, pag masyadong mura yung fish, di mo na kailangang i-vaccinate. Hayaan mo na lang mamatay, mag-culture ka na lang uli. But in terms of food safety, kasi may mga subclinical infections kasi tayo ng streptococcosis sa tilapia, and hindi natin malalaman by just looking at if the fish have infection or not. So we try to develop vaccines, and then we also have to do some health protocols, reduce the stock intensity, reduce the number of cages in Taal Lake. Pero these are very complex problems that involves a political and sociological aspects. So medyo mahirap. So vaccine, viable, pero ang uptake, if we use antibiotics, then there will be issue also on the use of antimicrobials. The streptococcosis can respond usually to antibiotics, but we will try to avoid the use of antibiotics, especially in aquaculture, especially in food fish.
Okay, sir, we understand. Thank you, sir. We have question from Lionel Lumugdang from Samas Malita, Davao Occidental for Dr. Obusan. Yes, ma'am. In Malita, we have 2 sightings of stranded dugong for 2020. What are the best practices that can we do in order to gather data? And hopefully we could participate in this study. Thanks. Thank you. Thank you, sir.
Hi, thank you for the information, sir. Actually, the best involvement uh, for you, if you're really interested, is to be part of the network. So the network uh, organizes training workshops for first responders and for those who can help us in collection of microbiological samples. But syempre, we are under community quarantine. Wala pa tayong face-to-face sessions or trainings. For now, you can visit the website of the Philippine Marine Mammal Stranding Network. Have there the different forms. So we have forms, for example, for reporting a stranding event. So in that form, we're asking the local stakeholders to provide data. So the appearance of the animal, the date and time of stranding, the particular site where the animal was found, and the names of the people who were there. And then in the website also, we have the different contact numbers. You can relay any message and we can give you guidance on how to properly approach the animal. But if you want the complete procedure, we have also a simplified protocol that can be performed by anyone who wants to help during stranding events. I can send you that paper, sir. So kung nakuha mo yung email ad ko, you can just email me and I will send you that paper. Nasa Davao ako, sir, last January 2020, wherein I presented also the microbiological findings involving those cetaceans that stranded in Davao region. So ay nasa ano kami last time, Ateneo de Davao. Thank you.
Thank you, ma'am. Another question po for you. What are the specific anthropogenic activities cause the presence of these emerging pathogens, since you've mentioned that these bacteria are initially found in the land and now that they can be found in the aquatic organisms?
Actually, in the case of Leptospira and Toxoplasma gondii, the hypothesis is that the pollution, biological pollution, is coming from effluents, from sewage, okay, from haphazard, discharge of untreated wastewater, for example, and there are actually researchers trying to model the pollution of our waters by seas, by Toxoplasma gondii based on the density of cats as the definitive host. So far, they have the report from US wherein they modeled the estimate loading of Toxoplasma gondii from different municipalities in association with the number of cats. Okay, so they have sampled wastewater and sewage effluents. A lot of anthropogenic factors are involved. In the case of antimicrobial resistance, we also have the use of antibiotics in aquaculture systems. So one of our projects actually is trying to associate the antibiotic resistance profiles to the types of antibiotic classes that are being used by our aquaculture farmers. But it's actually very difficult to, investigate a correlation because stranding events are random events. So we can't say that if we, detect a particular toxoplasma strain or a resistant bacterial isolate from this cetacean that stranded in call. So we can't say that the antimicrobial resistance is coming from Bicol as the geographical area, because some of our species are migrating from other countries and from other regions, and for most of our species we don't know their abundance, we don't know their ranges, we don't know their migration patterns. So ang dami-dami pang hindi natin alam about our marine mammals.
All right,
Thank you so
thank you,
much,
Dr.
Jack.
Busan. There are still a few questions, but unfortunately, that is all we have time for. So thank you to our speakers. Thank you to everyone for your questions. So the questions that were not answered today will be given to our speakers and they will respond to you by email. So again, thank you to our speakers, Ms. Joy Ann Santos, Professor Francis Legario, and Dr. Marie Christine Obusan for your engaging lectures. And thank you everyone for attending the webinar and for participating in the Q&A. We hope that you have learned a lot from the lectures this session. So please send us your feedback. The evaluation form is now posted on the chat box. So please evaluate in order to receive your certificate of attendance. So no evaluation, no certificate. Try to do this soon because the link will close 30 minutes after the end of the webinar.